Hi, my name's Rob Jones and I'm a consultant liver surgeon based in Liverpool, and I'm the co-lead of the NIHR UK Cancer Vaccine Innovation Platform.
And welcome. I'm Sarah Danson. I'm a medical oncologist from Sheffield, and I'm the co-clinical lead for the Vaccine Innovation Pathway with Rob.
So as part of the BGS webinar series, we're going to give you a talk on cancer vaccines for surgeons. And this is aimed to be a primer and a high-level overview of cancer vaccines for clinicians who are likely to be involved in trial and delivery. The objectives of the talk, we're going to briefly touch on the difference between prophylactic and therapeutic cancer vaccines. We're then going to give a touch on different types of cancer vaccine technology and how the different mechanisms work and the pros and cons of both. We're going to talk about how you decide what to vaccinate against. And then we're going to look at how we deliver cancer vaccines and how important surgeons are to this. And we're going to focus on the UK experience to date and a specific colorectal cancer vaccine trial as an example. And then finally, we're going to look at the role of surgeons in all of this. So very briefly, what's the difference between a prophylactic and a therapeutic cancer vaccine? Well, we're not going to spend too much time talking about prophylactic cancer vaccines, but we know that a lot of cancers have a clear etiology. So I'm a liver surgeon. I deal with a lot of hepatocellular cancer, and hepatocellular cancers tend to form in diseased livers. So vaccinations against hepatitis B, for example, reduces a pro-inflammatory oncogenic liver to reduce the risk of liver cancers. HPV is heavily implicated in head and neck cancers, and HPV vaccination can be a prophylactic mechanism to stop those cancers developing. There's also some really exciting work looking at prophylactic vaccines in high-risk populations. So the LynchVax trial, which is about to come out of Oxford, is looking at patients who have Lynch syndrome but don't have a confirmed diagnosis of cancer to see if we can vaccinate those patients to stop them developing cancers. The second group are therapeutic vaccines. These are vaccines where the vaccine is trying to target confirmed cancers. And that's looking at things like antigens which are involved in the cancer, or things like KRAS and CEA. So in broad terms, how do cancer vaccines work? Well, the principle is the same for any vaccine, really. You present something to the body and it's taken up by dendritic cells. So this purple cell here is an antigen presenting cell, and you'll remember that from medical school. So these are cells that take up antigens and they express them to the immune system in lymph nodes and the spleen. And what happens is your adaptive immune system comes to the dendritic cells and their antigens are presented, and it's then primed to look for the cells expressing these antigens. The T cells then move out into the periphery and they circulate around the body and they look for cells expressing those antigens, and then they destroy them. And that creates a circle, because by destroying those cancer cells, you then release more antigens. They're taken up by the adaptive immune system, and the cycle multiplies. There's lots of different types of cancer vaccine technologies, and today I'm just going to briefly touch on three of them: peptide and protein vaccines, cell-based vaccines, and then DNA and RNA vaccines. Probably the first question when you're making any kind of vaccine or cancer vaccine is, what do you actually vaccinate against? And whenever you're deciding what to vaccinate against, there's a degree of trade-off. And it's a trade-off between making something that's very, very specific to the tumour, so that the immune system doesn't think that it's a self-antigen and ignore it. So you need to have something that will trigger an immune response. The problem with getting stuff that is expressed on lots of different tumours and is well preserved is that that's often recognised as self by the immune system. So your immune system is very tolerant of it. But if you're making individualized tumor-specific antigens, then that becomes very complex, because it depends on the conserved antigens between cancers. And this is all a trade-off, and then we're going to go into that in some more detail. So peptide and protein vaccines, these are short fragments of target antigens, which are taken, peptides, proteins. They're safe, they're stable, they're relatively easy to produce. One good example is HER2 peptide antigens for breast cancer. The problem with that is that they're relatively poor at triggering an immune response from the immune system. And so a lot of the time, these need to be given in combination with other therapies to amplify the response. The other problem is that if you're only targeting one antigen, that the immune system could do something called immune escape, where cells start to change. They no longer express that antigen, and they can then no longer be receptive to the protein vaccines. Cell-based vaccines are really attractive. This is the idea of taking dendritic cells that are already primed to produce the antigens and present the antigens of interest to the immune system. And these trigger a hugely powerful immune response because they're getting straight onto the key points in the immune system. But they are fiendishly difficult to produce. They have to be individualized for each patient. They have huge complexity in transport and transfer and delivery. One example is the cilta-cel, which is used for metastatic prostate cancer. And it's shown great results, but it is really difficult to give. And then we're going to talk about DNA and RNA vaccines. These are customized. They take genetic material which is encoding tumor antigens, either tumor antigens which are expressed in all of the cancers or specific tumor antigens. And they're delivered into the patient, and the DNA and the RNA is coded into an antigen. It's expressed, and that triggers an immune response. And it's rapid, and it can be produced at scale. There are some issues with transport and stability. DNA also has issues with incorporation into the genome, and so there is growing interest there in RNA and mRNA technology. Specifically, a lot of the stuff which is spun out from the COVID-19 vaccines. So my own interest is in personalized mRNA cancer vaccines. So this is an example of the pipeline that BioNTech and Genentech use for their mRNA cancer vaccines. What happens is patients give blood and tumor, often taken by surgeons at the time of surgery, and rapid processing and quick storage of the tumor sample is absolutely critical to this. The tumor is then sequenced, and using a bioinformatic pipeline, we predict which neoantigens are going to trigger the greatest immune response. An RNA code for those antigens is then created and wrapped in a little lipophilic bubble that's quality controlled, and then that individualized, customized mRNA vaccine is delivered back to the patient. The vaccine is then injected intravenously, and these little lipophilic envelopes are taken up in an antigen presenting cell. And they transport the mRNA into the antigen presenting cell, and the mRNA is then coded into a protein, which is presented on the front of the antigen presenting cell and triggers the CD4 and CD8 cells, which then go off and track down any residual cancer cells. What are some of the main problems associated with therapeutic cancer vaccines? Well, the major one is immune tolerance to cancer vaccines. A lot of the things that we vaccinate against are recognized as self by the immune system. And if they're recognized as self, they're not going to trigger an immune reaction. Additionally, we know that the tumor microenvironment is hugely immunosuppressive. There's often Tregs and myeloid suppressor cells and interleukins, which dampen down the immune response, which means that the vaccine may be less effective, certainly against bulky and well-established tumors. And that's why there's a lot of interest in the adjuvant setting for minimal residual disease. There are lots of cancer vaccine trials that would be of interest to surgeons, and doubtless you will have read and heard about them during your reading and your training today. Our study at the moment, BioNTech 12201, is a colorectal cancer study. There's trials in melanoma, there's CEA-targeted vaccines in pancreas cancer, there's KRAS vaccines, looking at KRAS mutations in pancreatic cancer. Over 90% of pancreas cancers have a KRAS mutation. And what we're going to do is just look at a couple of examples.
So I'm going to talk about how we then take all of this technology and actually deliver it in terms of cancer vaccine trials. So talking about the UK experience. So as everyone knows, the UK led on COVID vaccine research. We learned about how COVID was as a disease, how it acted. We did treatment trials, and we also successfully developed vaccines over a very short time period. And we want to do the same for cancer vaccines. So the UK government has formed strategic partnerships with Moderna and BioNTech. So these are the two companies that produce most of the COVID vaccines. So the first of these partnerships was with Moderna, and this was signed in December 2022. And this is a 10-year partnership where Moderna has committed to investing in mRNA research and development in the UK. And the plan, which is now been brought through to fruition, is to build a state-of-the-art vaccine manufacturing centre with the ability to produce up to 250 million vaccines a year. These are vaccines for infectious diseases, for cancer, but it can also be used for other areas such as rare diseases. And then the second strategic partnership is with BioNTech, and this is specifically for messenger RNA cancer vaccine advancements, and that was signed in January of 2023. So this is looking at the BioNTech cancer portfolio, and as you can see on the left-hand side, we have the mRNA cancer vaccines, which are what we're interested in here, and this includes both individualized cancer vaccines, so specific for that individual tumour, for that individual patient, but also what we call FixVax, which is where we take a vaccine off the shelf and target it against the common abnormalities in that type of tumour. And so we've got a variety of trials going on with both of those strategies. But you can see that BioNTech also brings these other ways of treating cancer with them. So we have cell therapies, antibodies, small molecule inhibitors, next generation immune modulators, and the ribologicals as well. So just picking out one of those on the left-hand side again, with the cell therapies, we've got CAR-T coming through. Now, this is something where we take out a sample of tumour. We also are taking out the body's immune cells against that tumour. We increase the number of those immune cells outside of the body. We then prime the patient's body by reducing their immune system in the first place with chemotherapy and then priming it with immunotherapy. And then we reinfuse those immune cells into the body. And the idea is that they then work in a much magnified way against any tumour cells. So that's something that BioNTech's bringing as well. And you can see at the bottom that that the UK BioNTech partnership goal is for us to treat up to 10,000 United Kingdom patients with personalised messenger RNA immunotherapies by 2030. So big goals, which is why we've set up the infrastructure in order to deliver this. And this is the equivalent for Moderna. So you presented in a different way, but you can see that Moderna has infectious disease vaccines. That's a large part of their portfolio. They have the cancer vaccines and therapeutics, but also they've got regenerative therapeutics, cell surface therapeutics, and inhaled respiratory therapeutics as well. So looking at the dark blue area, we have those individualized cancer vaccines that we've talked about. We have checkpoint vaccines as well, which is part of the vaccine innovation pathway. We're targeting mutations in the tumour, such as KRAS, and there's lots more research going on into expanding this area as well. So it's a huge pipeline. So in order to deliver this, this was a timeline as to how the UK has progressed things. So as you know, in March 2020, the World Health Organization declared the COVID-19 pandemic. By December 2020, we had the first COVID vaccine in place. So very short timeline. By December 2022, we'd signed that Moderna strategic partnership with the UK government, and the following month, the BioNTech strategic partnership was signed. In February 2023, the UK vaccine innovation pathway was launched in order to deliver on these two strategic partnerships, but also to expand out to other companies as well. And in June 2023, the Vaccine Innovation Pathway Programme Management Office was established. So this was a team of people working full-time in order to deliver on this research. In November 2023, in parallel with the O'Shaughnessy report, which was looking at successful delivery of UK commercial trials, the Vaccine Innovation Pathway was designated as one of two clinical trial delivery accelerators. So this is top priority for the government. That was why it was made a clinical trials delivery accelerator, to be at the forefront of everyone's mind. And by September 2024, we were really working on accelerating trial setup timelines. So this is an infectious disease trial that's mentioned here, which was set up within 8 days from UK regulatory approval. So this is what we need to be doing, is getting the trials and opening them as soon as possible. This is just to say the team that's behind that, so that which is ever increasing. So we've got Maria Kufali, who at the top, who's the head of the Vaccine Innovation Pathway. We've got myself then as one of the co-clinical leads, Pippa Corrie, who was working with me from the beginning as the other co-clinical lead, and then Rob took over from her point of view. So we have set up a UK Cancer Vaccines Research Forum to complement what's happening with the Vaccine Innovation Pathway Office. This is so that we have research-ready investigators and sites. This is co-chaired by David Cameron from Edinburgh and Chris Twells from Leeds. And the idea is that we are able to provide expert advice to sponsors, the NHS, and also the NIHR. It allows us to contribute very effectively to the NIHR response function for swift resolution of emerging issues from these cancer vaccine trials. And what we really want is to see these protocols early on, so that we can actually say if we can deliver on these trials in the United Kingdom using our pathways that are in place. And what's really important is that we're training people as we go along, such as with this lecture. So we're supporting training and education requirements for delivering these agents. The people on this research forum include principal investigators, the people from the Cancer Vaccine Launchpad, which I'll discuss next, ECMCs, that's our experimental cancer medicine centres. So those are the ones that deliver on the very early phase trials. Biomedical research centres, where lots of the intelligence and the research development goes on. Clinical research facilities, which deliver on some early phase trials, but also the later, bigger trials. Clinical trials units, which help us design and deliver trials. Our PPI, so our patient and public involvement. And also, this is not just about England, this is about the whole UK. So we have representatives from the devolved nations as well. So we have our cancer vaccine launchpad, which is something that was put in place, first of all, for the colorectal trial, which Rob will talk further about shortly. So this is where we're identifying patients whilst they're going through the pathway of having their surgery for colorectal cancer. We're logging them into the system, taking blood samples to see if they have circulating tumour DNA, which we think is a first pointer of when their disease is more active. And then we're monitoring them through the system. If they are eligible for the trial, so if they've got circulating tumour DNA, then they can then be identified and hopefully as many of them as possible put onto the trial. But the great thing about this pathway is that is not just colorectal cancer specific. So we're now expanding this out. So we've already brought in a head and neck trial that we're using this for, and there's just been a lot of publicity about the fact that we're now going to use this for a melanoma trial as well. So we hope that eventually we'll be screening more and more patients in order to get us to those high numbers that we want to deliver through the vaccine innovation pathway. So we need to recognise at this point that there are challenges to delivering on cancer vaccine trials. They're very different from many of the other trials that we do. Initially, there were some setup delays because you've got many different types of individuals and support services involved in delivering each of these trials. So you need pharmacy to be involved, you need radiology to be involved, you need surgeons as well as oncologists to be delivering these. So we've had to work through the pathways to try and do these as effectively as possible. We've learned that you need a sustainable infrastructure with protected clinician time, but also with nurses that aren't going to be pulled off to do other jobs. And you need coordinators in order to put all of those teams together. If you're going to deliver on these things effectively, you need to have space to do it. So that might be having the chair capacity in order to deliver the cancer vaccines, or with the infectious diseases, it might be having the large spaces where you can actually get people as quickly as possible to come through. And this also encompasses things like remote delivery vehicles as well. So you may have a van that goes out and gives the infectious disease vaccines. Unlike the infectious disease vaccines, the cancer vaccines have this classification. They're called ATIMPs, so that's Advanced Therapeutic Medicinal Products. And that classification means that they do have to be treated in a slightly different way than the COVID vaccines. So pharmacy need to log them in a different way, but also prepare them, sometimes in a fume hood. So we need to make sure that pharmacists have the space in order to do that, and also the familiarity with doing it. So that brings me on to the fact that there are specific vaccines and immunotherapy skills. You don't just have to be able to deliver the vaccines, you then need to be able to treat any side effects or problems that come with it. So people are learning very rapidly about how to do this. And finally, what we need is we do need to make sure that this is not something that's just given in a few centres around the UK. So we want every patient that's got a particular type of cancer to have access to cancer vaccines. So this is where participant diversity and inclusion comes in, and we work with our patient and public in order to be able to do that. We've learned a lot of lessons from COVID vaccine research in order to do that. So here we have an example of how we're trying to help the community to deliver things more effectively. So this is a document that we put together for pharmacies, talking through the key considerations as to how they deliver cancer vaccines most effectively. So the idea here is that each site is using what others have already learned rather than starting from scratch in order to be able to deliver things. And part of this is about us making sure that everyone knows what's going on with cancer vaccines. So the UK has done a huge amount in order to get this recognition. We've had a Grand UK Cancer Vaccine Summit, and we're due to have a second one of those next month. There have been many news articles about cancer vaccines on the television, on the radio, in manuscripts, but also podcasts as well. So this here is one example. So this was in the British Medical Journal, which many people, of course, read. So it's about the rapid rise of the UK's cancer vaccine trials, how we are very much trying to position ourselves at the forefront of that. So we've delivered really well to the colorectal cancer trial, but also the study that was mentioned, the Moderna study 4157 in melanoma and lung cancer. We've recruited a third of the patients globally into that. So we'll continue to do this type of dissemination in order that patients and clinicians know what is available out there.
So for this final section, we're going to look at a trial that I was heavily involved in as an example of whether these models work and how surgeons can deliver cancer vaccines. I know not everybody here is a colorectal surgeon, but BNT12201 was a trial looking at adjuvant treatment in colorectal cancer. So the question the trial asked was whether you can improve the survival for patients after they've had surgery by adding a cancer vaccine to standard adjuvant chemotherapy. And it was looking at disease-free survival. It was taking patients with resected high-risk stage 2 or stage 3 colorectal cancer, so the group who would normally get adjuvant chemotherapy as their standard of care. We were interested in patients who were ctDNA positive. Why was that? Well, this was a study called the Galaxy trial from Japan, and it looked at the survival for patients after their colorectal cancer surgery based on their ctDNA positivity at 4 weeks. And they found that if you were ctDNA positive after you'd had your primary removed, your survival was far worse, a hazard ratio of 10. And that makes complete sense because you've got occult micrometastases that we don't know about. So we were interested in this group. They had small volume residual disease. And once we got that group, we then wanted to make sure we could make a vaccine. Once they'd finished their chemotherapy, we randomized them, and we needed 232 patients. And actually, those 3 screening steps meant it was only 6% of patients that we screened for the trial could we put through to randomization. So we had to work really hard to go out and find these patients. So we had a few different models. In Liverpool, we did something called a hub and spoke system. All of our chemotherapy is delivered by one trust, Clatterbridge, but they run multiple satellite sites. So we screen patients at all of these satellite sites for eligibility. And only the patients that were eligible did we ask to travel to the main vaccination centre. So we used a filtering model. The second thing we did a lot of was something called a PIC Plus, Patient Identification Centre Plus. So what would normally happen is a patient would have their surgery, they've been seeing a surgical outpatients afterwards, and maybe the surgical team would mention about the vaccine trial. And then when the patient went to the trial site, they'd see the oncologist and they'd get some information, they'd go away and have a think. And then if they were interested in taking part, they'd come back, they'd be consented and they'd do a blood test. We changed that from just mentioning the trial to actually advertising this trial to patients before they had surgery. With CT scanning and MRI, you can often pretty accurately stage these colorectal cancers before you get the pathology. And so we had a good feel which patients were likely to be eligible. Then once they came back after surgery, the surgeons gave them the information sheet and taught them about the trial. And that really built on the relationship that you build with your patients when you've taken them through a major operation. The patients were then very receptive to the idea of taking part, and they were contacted before they went to oncology about the study. And then a research team member was able to see the patient and consent them for the blood test before they even went into their oncology outpatient. So the oncology outpatient could just focus on the important standard of care discussions about whether they should have adjuvant chemotherapy or not. Sarah talked very elegantly about the NHS cancer vaccine launchpad, and this was the idea that it doesn't matter which hospital you present at and whether there's a vaccine trial open at your hospital, you should still be able to access it. And it's a huge NHSE activity. It's been hugely successful. If we just relied on our 14 trial sites, we would only capture the patients who were going to those 14 trial sites. But the CVLP means that we're able to capture all of the other sites near these trial sites. So our geographical coverage across England, if you look at the map there, is absolutely enormous. The other thing that we've done is mobile capacity. We realised a lot of the time we're asking patients to travel a long way for lots and lots of clinic appointments just to see if they're eligible to take part. And that's a real turn-off. And so what we've done is we've started to use some mobile capacity where we take the trial and the screening processes out in a van to satellite centres, and we actually screen and recruit the patients using these. And then only when patients have got the carrot of getting a potential vaccine do we ask them to travel to the main trial site. And that's really important for making sure that the patient population who we get into our trials represent the patient population that we treat week in, week out. So does any of this actually work? Well, it does. It clearly does. This is the recruitment graph for the BNT122 trial across the globe, and you can see the UK recruitment is the orange curve, and the models that we've used and the UK cancer vaccine infrastructure means we are global leaders in terms of recruitment for this trial, and we're likely to be the same for all other future cancer vaccine trials. We recruited over 800 patients. That was 44% of the global total over the last 6 months, 40% in the last 12 months, and we've only got 14% of global sites. So these models really mean that we can punch above our weight. Because of the cancer vaccine launchpad, we're actually able to offer 60% of UK patients access to this trial if they wanted to take part, which is phenomenal. You know, if we relied on just our traditional site model, only 15% of UK patients would be eligible. But because of these models, we've offered real diversity of access and equity of access, and over 60% of patients are now able to access the trial. So it's been a real success story. I just want to briefly touch now on what the surgeon's role in cancer vaccine trial delivery and development can be. The first thing is that we've taken patients through what can often be a very, very difficult operation and recovery. We have a strong relationship, an emotional bond with them. If we talk to them about a trial and we think it's of value, they're going to think it's of value as well. And so that first point of contact can really influence whether people decide to take part or not. The second thing is our role in biospecimen design and delivery. We know that for anything involving sequencing, tumor stability and preservation is fundamental. And so one of the changes we made in the colorectal study was we used to get our surgeons to open the anti-tumoral border of the bowel, clear from the margin, to allow formalin into the specimen immediately after it was removed. And that stability made a massive uptick in our tumour retrieval and our ability to generate high quality vaccines. Surgeons know what happens to patients before, during, and after surgery, and so our input into clinical trial design, how it will align with standards of care, and what the clinical need is absolutely fundamental. And finally, surgeons are good at recruiting. It's been suggested we may be a slightly competitive bunch. And so if we know that somebody in the hospital down the road's recruited 10 patients, we're going to want to recruit 15. And that really can power on our vaccine delivery, our vaccine recruitment and delivery trials. So in summary, cancer vaccines are a rapidly progressing field. It's hugely exciting. There's a lot going on. Solid organ cancers remain the main focus. The UK is a global leader now in terms of cancer vaccine trial design and delivery. And surgeons are absolutely pivotal for the leadership, for the recruitment, and the delivery of these studies. Thank you very much for your time.